In this activity, you will examine the internal structure of a monocot leaf using a prepared cross-section of corn, via maize. Externally, monocot leaves look simple and narrow, but internally, they contain highly specialized tissues organized to support photosynthesis, water transport, and protection. You will identify the epidermis, mesophyll, and vascular bundles and observe how their arrangement reflects the functional needs of the leaf. Internally, monocot leaves are composed of three major tissue systems. If you notice, the upper and lower monocot leaf is nearly identical. This is an adaptation for leaves that grow vertically, receiving sunlight equally on both sides. The epidermis forms the protective outer layers and contains stomata, guard cells, and sometimes specialized cells such as trichomes or bulliform cells. Bulliform cells help minimize water loss by curling the leaf when it is dry. The mesophyll is the photosynthetic ground tissue made primarily of chlorenchyma cells rich in chloroplasts. The vascular bundles, or veins, contain xylem and phloem surrounded by a bundle sheath. These tissues transport water, minerals, and sugars and help limit water loss. In many monocots, the xylem is positioned closer to the upper epidermis while the phloem lies closer to the lower epidermis. Having the xylem closer to the upper epidermis allows efficient delivery of water and minerals to photosynthetic tissues, while the phloem moves the sugars created away from the leaves. Before you begin observing the corn leaf cross-section, take a moment to think about this question. Why is the xylem positioned closer to the upper surface and the phloem closer to the lower surface in most monocot leaves? And how does this arrangement support photosynthesis and transport? As you form your prediction, focus on what each tissue moves. Xylem delivers water and minerals, and phloem transports sugars made during photosynthesis. Now connect that to where photosynthesis is happening in the leaf and where materials need to go. Pause the video and record your prediction in your lab notebook. We'll begin by viewing the corn leaf cross-section under low power. Locate the overall outline of the leaf and identify the upper and lower surfaces. Scan across the section to find several veins, and then locate the largest one in the center, the midvein. Take a look at the corn leaf on the right. In the photo, you can see the large midvein and the parallel veins. You can see that same pattern in the slide. Since monocots have long, narrow leaves, all those veins running parallel are essential to transport water and nutrients and provide that structural support to prevent the leaves from tearing in high winds. Now focus on the epidermal layers. Identify the upper epidermis and lower epidermis. Look for stomata and guard cells. You'll notice an air pocket just inside the stomata, although there are not stomata where all the air pockets are. Which surface appears to contain more stomata? Although this view may show a few more stomata on the lower side, in general, monocot leaves have roughly equal numbers of stomata on both sides. This is a characteristic feature of monocots to allow more efficient gas exchange since the leaves are typically upright rather than flat. Next, locate the mesophyll between the two epidermal layers. Observe the size, shape, and packing of the chlorenchyma cells. Do they appear loosely arranged or tightly packed? Do you see air spaces? Is there any obvious layering, or is the tissue fairly uniform? In monocots, the mesophyll consists of uniform chlorophyll-containing chlorenchyma cells. We'll see different types of mesophyll in eudicots. Now examine the midvein more closely. Identify the xylem, usually with larger, thick-walled cells toward the upper surface, and the phloem composed of smaller, thinner-walled cells toward the lower surface. How can you visually distinguish xylem from phloem based on position, cell size, and wall thickness? Locate the bundle sheath surrounding the vascular tissues. The midrib will have a more pronounced bundle sheath due to its extension to the upper and lower surfaces. Each bundle is typically surrounded by sclerenchyma as well as parenchyma cells with a higher amount of chloroplasts called Cran's anatomy, which allows for more efficient photosynthesis in C4 plants. You'll learn more about this in the photosynthesis section. In some monocot leaves, especially grasses, the upper epidermis contains bulliform cells. These are large, thin-walled cells that lose turgor during drought, causing the leaf to curl inward and reduce water loss. Look for unusually large, bubble-like cells in the upper epidermis. Now create a detailed label drawing of the corn leaf cross-section in your lab notebook. Include and label the upper and lower epidermis, mesophyll, xylem and phloem, bundle sheath and bundle sheath extension, and bulliform cells. Pause the video while you complete your drawing. Here are some compare and contrast questions. How does the lower epidermis differ from the upper epidermis? For the first question, compare stomatal abundance, cell shape, and any specialized cells you observed on each surface. Describe two differences between the midvein and smaller lateral veins in the leaf. For the second question, think about the relative size of vascular tissue, amount of supporting tissue, thickness of the bundle sheath, and overall function of transport versus structural support. Write your responses in your lab notebook. To summarize this activity, monocot leaves contain specialized tissue systems organized for efficient photosynthesis, transport, and water conservation. The epidermis regulates exchange with the environment, the mesophyll performs photosynthesis, and the vascular bundles distribute water and sugars while providing structural support. Bundle sheaths and bulliform cells illustrate how even individual cell types contribute to whole leaf function. Once again, the organization of tissues reinforce the central theme of plant anatomy: structure supports function.